Hello everyone, welcome back to my channel. Welcome back to Ming Daily. And if you haven't watched the previous episode of me going to Macclesfield, I suggest you head there right now. I don't know, it's gonna be somewhere. In today's video in this video I'll be giving you sort of a summary as well as a reflection, a daily reflection of my first week interning at AstraZeneca. So before starting this internship, I've done a few internships in the past, but before starting this internship I felt like I wanted to make a really good record of what I did here, what I've learned here, because I think that it is finally an internship that's aligned with my future career goals. And I feel like in my future applications to write something that is, not to say really convincing, but something that can maybe stand out, or an experience that is quite unique of my own, is definitely this one that I would definitely want to touch on. So I don't want to forget any moment of it, and hence this video log. I will every day, so I'll do it by week, so each series will be by week. Hopefully it won't be too long.
It was at this moment that she knew she fucked up.
This is reflection of day one working at AstraZeneca. We basically had an induction this morning. We're supposed to report in at 9, but everything got delayed a little bit. But the induction was basically on safety, health, and environmental. So, of course, working in a lab, safety is very important, your health is also very important, and the environment, they want it to be a very energizing environment. So, and with that, there's a little pamphlet here. It is my golden rules. Might be flipped in your camera, but yeah, like the rules that AstraZeneca stand by on, that we should obey all the time. And a very, very good quote from them, which is, there is nothing to rush that you can't do it in the safe way. Something like that, which I think is really important. Environmental, so what things can have some impact on it. As well as welcome to the Macclesfield campus, because if you haven't checked out my previous video, it is huge. Yes, I mean huge. And they didn't tell you how to get to reception on the first day. But yeah, that's a bit of the map. And apparently, I also just learned today, very stupid of me, is that AstraZeneca, I mean, of course, it's a merger, Astra and Zeneca, and it's actually a British-Swedish company. So, whilst there are a few research centers in the UK and manufacturing centers, a lot of the research work and manufacturing work are in Sweden. So my manager was telling me about how we have a research unit on oral product development unit in the UK in Macclesfield, and there are probably 3 or 4 more in Gothenburg in Sweden. So yeah, and they get to fly to Sweden a lot, which is my dream. So if I land in AstraZeneca, I might pray that, oh, can I go and work in your Swedish branch? Anyways, yeah, so after that, so we had introduction, but that comes later. So after briefing, of course, I got my laptop. I got a whole keyboard. Don't think I use it for my work laptop. I'll use it for my own laptop because my laptop keys are not working. And then what do we have? Introduction. So basically, it's, I had to read SOPs, but I cannot access the website yet. So that will have to take a pause. But I had a read through on what I'm gonna do in my project, and I've written down a bit. I'm not sure if I can share everything with you, so let's just see what I can share and what I thought about it. So basically, okay, so I think this one I definitely can share. So basically, as per my introduction with my line manager, is that AstraZeneca, whilst their team is doing product development on obviously new medicines, on the side they also do sort of like sort of things to improve the whole flow. So either improvement methods on how to not calibrate, what I'm talking about, improvement methods on how to sort of it's not coming to me, like when you have to measure something and you're using like a better equipment or a better process so that it's more efficient, so that it's more accurate. So that are things that they're improving on, but clearly they don't always have the time. They have to produce new products. So that's where students come in, because again, it is it's still important, but it's not that serious if it doesn't work out well, etc. So what I have to do is to develop a calibration model using I'm not sure if it's a new equipment, but a different equipment that they normally use for a medicine. And unfortunately that medicine was discontinued, but they really want to create this calibration model detection method so that in the short term we can check for why we had to discontinue it, like what's the reason for causing the irregular content uniformity, whereas in the long term, for other medicines we can use it as a quick like detection or an end process check, instead of like for the QC, the quality control team, to go through this whole long process of just checking if there's like uniform content. So with that, I'll also be learning some new skills using different equipments that they have. And then I was reading on like some of the background work that they were doing. So they have done like biopharmaceutics and physical chemical properties of the API, the active pharmaceutical ingredient itself. And like what are some of the data they got from patients. And then after that, my line manager, before he left work, he brought me to their labs for a tour, and my gosh, they have a lot of equipment, a lot of high-tech machines. I've listed a few here, but I don't even think these are the ones I can remember on top of my head, and I think these are quite normal because I've heard them before. It's like DSC, differential scanning calorimetry for thermal analysis of the product, scanning electron microscope, so obviously for imaging, static light scattering with laser SLS, that's to test the particle size of powder. So they have a lot of work on material science as well as solid state. So solid state characteristics are on things like the crystallization, so their structure, morphology. So if it's like an amorphous form, a hydrated form, a hemi-hydrated form, so like how many water molecules it ties together. So they have also of these things. They have annular shear cell to measure powder flow, X-ray diffraction, all sorts for imaging. They also have things to measure hygroscopicity and wettability. So all sorts of things that we read. Like I used to study in pharmaceutics, but not go in depth, obviously, because it probably takes years to master the skill of using these. But these are all the equipment that AstraZeneca and probably a lot of pharmaceutical companies invest in. And also he showed me on like the new drug application process, like all the details that you need to file in. It's like I don't know, a few hundreds or 1,000 pages of words. So there's a lot of work being done in for a new product to come out. Although they do earn a lot of money and all the medicines are very expensive, it's really like, how do you say, it's really something that is they really put a lot of time and effort and manpower into on their research. They basically think of it like, in their research, they don't just like try out one way of getting the final product out. They also have to test for all sorts of possibilities that could go wrong, and what are the sort of like maybe the parameters that must be very strict. They test all of them out and then write them out in their method control, so that even in the case, like in the worst case scenario, the patients are not severely affected. So yeah, I would say that's day one. Obviously day one has definitely a lot more things to talk about. It's around 9 minutes plus now, so I'm going to stop speaking and I'll see you tomorrow. Bye. Okay, day 2. Well, today's a very busy day. I'm very tired, so if you don't mind, I'm going to be removing my makeup whilst I give my evaluation. So basically what happened today was I actually went in the office a bit earlier, but I had to sort out my visa application, so that was a nightmare. I went in at around 8, but only actually started work at 9 when I had to attend a meeting, and after that was just back-to-back meetings. So I had 3 back-to-back meetings, lunch, and then another meeting. So that was great. So the first meeting was an introduction to the pharmaceutical technology and development team, which was nice and simple. I think it so he really the line manager, but not my line manager, another intern's line manager, but they're all like head, right? So he basically gave us an overview of the whole company structure and like what are the different stages that, you know, a medicine has to go through before they can be approved, which is really long. Like, oh, so the first thing he gave us like a bit of like a quiz, right? So every day globally, AstraZeneca spends 26.6 million just on research and development. So that's not including like financing, marketing, sales. Every day they're already spending 26.6 million. So that's why, you know, like, although medicines are expensive, they're not charged like without reason. Yeah. And then yeah, so basically we talk about like the process, and then how now there are more medicines going into like personalized medicines, and how, you know, it will impact pharmaceutical companies, right? So but I guess there is some way that they can do, so they can start manufacturing the material such that it's a very simple process for the pharmacist in the pharmacy to quickly formulate the final product according to the patient's needs. And I think this is something quite interesting. So yeah, that looks like something interesting that we can definitely look forward to in the future. And then the next call, I was in a call with my project team. So the project that I'm doing, basically I'm developing like a calibration model. So the whole project, they were sharing like how it came about, what are some of the key timelines. So that was really nice. So in my team, I mean my supervisor is obviously from Macclesfield, and there's another guy who's also from Macclesfield, and then another guy from Sweden. Yeah, he's based in Gothenburg. And then there's another one who was actually my interviewer, so she's also on the team, but I'm not quite sure her role. I guess she's supporting with the because she was in the piloting project of manufacturing this medicine, right, until it got discontinued. So yeah, that was that. And then I was supposed to have my occupational health check, but they didn't show up. And then I was like figuring out my visa stuff, and then it didn't work out until after lunch. When I get on my computer, it started to work. So, but yeah, not really work related. And after that, I read again on my project, so more details on my project, and I also started looking into the different machines. So there was one that was like the Boxluck Turner. So that's to blend the powder mixture, and then I'm also going to use something to encapsulate, make them into capsules, and then only test them on the spectroscopy. So that's what I have to do. And then we also had like another health and safety induction today by another line manager. So that one, I feel like everyone was really sleepy by that point. Our SOPs were also just released, so everyone was also busy reading SOPs, but I haven't started mine yet. I will start them tomorrow probably. Yeah, so that second day, another long day. Okay, not as long as first day. But yeah, I think that's a good summary of what happened. I think what I learned is that there are indeed a lot of expertise that are involved. I know in like pharmacy degree we were taught of like the different stages of like formulating a medicine, not just formulating but coming up. So you know how you discover the molecule, discover the receptor side, and then all the different stages of sort of modifying the molecule so that it has better affinity, it has better solubility, etc., just to make life easier for the formulators, right, when they create the final tablets or capsules. So yeah, actually there are a lot of people in the team, and you can't do the work all by yourself because it's just impossible. And yeah, so I guess that just shows like everyone has their own specialty in the whole project. So that's why they like in a pharmaceutical team or in a pharmaceutical industry, you'll have people from many different expertise working together, working as a team, and everyone is important, no one's not important. And I guess communication is really important in that aspect, because when you're focused on you're basically doing a research to prove just one tiny element of that whole big process of a medicine. Yeah, so definitely a lot of work goes into these medicines, so we should always cherish them and really not take them for granted. I guess that's all for day 2. I'll see you back tomorrow at day 3. Bye. So it's like 7. It's a bit late now, so I thought I'd just do my day 3 summary on the road whilst I'm walking back. And sorry if you keep seeing me wearing the same shirt. I do change. I just went to the gym, so I'm wearing back the same shirt. And if the videos are wobbly, that's because my legs are barely hanging on, so I'm so sorry about that. So yeah, day 3, fairly simple. I spent a lot of today reading through the SOPs, some safety trainings, and then after that we also have a very detailed like 2-hour safety training that's on like using the labs, what are the substances that are hazardous for use and how we should use them, such as like using them in fume hoods, not learning how to use the fume hoods, and also what else, like there are some areas like with negative pressure, positive pressure, can't really remember, yeah, and like the PPE or what types of equipment to use, and the different categories of the substances. So depending on that, it's like how much caution we should take. So that's what we did in that 2-hour session. It was lunch, and I had a tour of the building that I will be working in. So I had a tour in the building that I'll be working in, and it was so cool just to see like tableting machines, capsule machines, granulating machines, and also machines you could use to sort of make them into ribbons, so that what do they call it? Rotationary. It's sort of like compress all the powder beds into a ribbon, and then you break it down into granules. So it's better for making that for filling them. It's like giving them a better flow to fill them into capsules. I feel like, oh my god, all the theory that I've learned actually comes like it's used, whereas in the past I'm just like, I don't know why I'm studying this. And you're actually having to see all the machines. Even our machines are like lab based, so it's not as big. It's definitely bigger than the ones you get in universities, but the formulation scientist that was bringing me around told me that in the manufacturing plant it's way bigger and way faster. So yeah, I don't know how much of the inside I can take a video, but yeah, that's the summary for day 3, and then see you tomorrow for day 4. Bye bye. Hello. So this is day 4 of me working at AstraZeneca. Apologies if you suddenly hear car noises or people's voices. My room's facing the road and it's just a bit noisy. Okay, so what I did today, a big chunk of my time today was again spent reading SOPs. But I actually found one that, not found one, but like the SOPs are now becoming more relevant to what I'll be doing, so like lab-based testing. So I found one which was really interesting, and hence I've written them down, which is why I also wanted to sort of share with you a little bit, because I think this is just an SOP, which is perfectly fine. So actually this SOP is on stability studies. So there are 3 main factors that affect things solubility, so like drug and excipient formulation and the environment. So obviously the API itself and the excipients, they have different chemical structures, solid state, moisture content, etc. So the different factors, and within the formulation, so their interaction with each other, as well as the process basically, and also packing up powder. So how would they, like all the different temperatures, etc. Actually no, that will fall under environment. So temperature, humidity, light, oxygen in the packaging would affect the stability of the drug. So obviously with stability tests, you do some accelerated tests, which is to either increase humidity, increase time, increase temperature, and with a set storage time, like test out how much of the API has degraded, and what's the what would it factor out to be like the shelf life of the medicine, obviously. So, and then for that they did dissolution test, impurity test, and potentially a different climate test, which means that if you go to if you work in like if you send out the medicine to a different climate, like if you send out the medicine to Africa or Asia, they have different climates, and how would that affect the stability of the medicine itself, not just the API, but also the excipients or the whole formulated drug itself. Then there are some methods of analysis. So They measure the degraded products to see if they have increased or decreased, the disintegration, dissolution rate of the products in the solid state. So if they change phases, that means that it may affect like either its solubility in the body, bioavailability in the body, or it can degrade faster. So, and then in the appendix, there are some main degradation pathways. So obviously thermolytic degradation with heat, hydrolytic, technically a part of thermolytic, but because it's so major that they split out, oxidative, and yeah, and also a few which are kind of new are process-induced transformation. So some of these excipients actually, they induce these phase transformation. Yeah, so something to take note of, like when using these excipients. And deliquescence is something new that I've learned of, which means that it speeds up like the rate of melting or dissolving because the drug absorbs moisture from the air. So that was one interesting SOP that I found out that I thought I would share because I found it really useful, and hence I wrote it down. Yeah, and then after that we had a training on the electronic laboratory notebook. Basically, you know how we always write down our experiments on paper, or we type it out in a Word document, etc. Basically, in AstraZeneca, and I'm pretty sure with other companies as well, they have a system of logging in all this information so that it feeds into a larger database. And if there's like a quality control and auditing, they can easily pull out all this information, because as I've mentioned earlier, like in the previous videos, which are that in one new drug application document, there are so many tests that needs to be carried out, and it's not just one person carrying out all the tests, but tests being carried out by different specialists. So everyone would log in these information to the database, and when they need to produce the final NDA, or if someone wants to review like the medicine, or if something goes wrong and they want to check what's wrong with it, or if they want to replicate the process and they want to figure out what's the best process to work on it, that's where they would go into this database to access all the information. So that's just some information that I've written down. And then at the end, at the evening, so because I did that after lunch, and then for the rest of the evening I did a test sort of. So yesterday we had a training on like safety and hazards, so we had to complete the test, and if we pass it we can only enter the lab. So hopefully everything goes well. And yeah, I was also scheduled in with a few last minute meetings for tomorrow, which is Friday, and one of them is actually a check-in with my manager. So I've also been spending the afternoon just reviewing my week, what I've done, what went well, you know, what I've learned, what I would like to improve on, and what are some of the goals that I would like to achieve in the next few weeks since learning more about this project. So yeah, we'll see how that check-in process goes tomorrow. I'll keep you updated. See ya! Hello! So this is day 5 of working in AstraZeneca, and today has been actually very, very interesting. So we have kind of like a flexible working hour. So technically you work from 9 to 5, but you can also work earlier, like 8 to 4. Today I actually went into the office super early, like at 7 to 3, because I need to leave to Manchester in the evening, but that's a story for another time. So basically I went in at 7. My laptop was malfunctioning and there was no one there, so I just figured it out myself. And then I started reading some SOPs, and then by 9, we had a lab tour. So I basically was on the tour before, but there were extra health and safety information being given to us. And after that, I actually went to collect my materials that I need for my project. So it was a lot of weighing out the powders that I need, and weighing out the excipients, the API that I need for the analysis. Like probably my first time working in a lab there, and we were actually using the not just a regular fume cupboard, but they call it like a powder cabinet, which means it's designed specifically for handling powders. So the filter is more suited to sort of prevent powder from flowing out, mainly the API, because it might be toxic, or if you don't handle it properly. And then so yeah, that's what I did until lunchtime. And then in lunchtime during that talk I was with my supervisor all the time in the lab. So it was really nice. We were just like chatting about career progression, like what she has done since graduating, because she's also a pharmacist, but she became like a senior scientist now. And after that, we scheduled a meeting with a recent graduate. So he also underwent the summer placement at AstraZeneca, and then he went into pre-registration with AstraZeneca, and now he's in the AstraZeneca graduate program, which I don't know, cross my fingers, hopefully I can be like him. But that was really, really cool because I learned about his experiences, and he also gave us a lot of context of people that we can reach out to within AstraZeneca, just to learn about their career journey, how they evolve, because I personally have quite a big passion for many things. So I like formulation. I'm also quite interested in like biology, pharmacology kind of things. So I was thinking like, oh, maybe I want to talk to someone who is working in the early stage development. So that's what I might do. Reach out to them. And after that, my manager had a first week check-in call with me, which at first I was really, really nervous, but it turns out to be not that so-called stressful or tense. It's basically literally just him checking in me to see how I'm settling in, what I'm doing right now, if there's any problems. So yeah, I think that was a good first week summary. I felt very welcome in AstraZeneca. I love having coffee and tea breaks or lunch breaks with the staff, just talking to them. Everyone has such amazing background and everyone is so knowledgeable that you just learn so much. So definitely, if you have a chance to go into any internship, talk to the people, because you think that, oh, they only know this much, but actually they know loads. And even if they don't, they can direct you to people who can give you these information. So yeah, just keep your options open. That's all I can say. And then just pursue things that you like to do, because there's always a route for you to do things that you like to do. Don't feel that you need to fit into one mold or the other. And I think for sure your passion will shine through if you go into interview, etc. So be well prepared, of course, but don't be too stressed out about it. If you're fit for the job, you get the job. What was I talking about? So yeah, that was day 5, and that concludes up my whole first week in AstraZeneca. Wow, it was so crazy, isn't it? But thank you so much for watching this mini series of my internship experience in AstraZeneca, and I will see you next week. Bye. Also, drop down in the comments if you have any questions about basically AstraZeneca, my experience here, I'll try to or if there's any information that you want to find out, that if it's suitable, I'll try to ask around for you. And yeah, so drop them down in the comments below. If you enjoyed this video, please subscribe, because yes, there will be hopefully 9 or 10 weeks worth of these videos, and hopefully I'll be able to sort of share as much of my experience as I can with you, and also to answer any of your questions, because I understand that it's really hard to step into pharmaceutical industry, and I know that I'm really grateful for this opportunity that I have right now, and if possible, I would like to share them as much as possible to all of you who are also looking to venture into pharmaceutical industry. So yeah, it's not that scary as you think, but you do need to be really good at what you do. Yeah, sorry. Just drop any questions in the comments below. Subscribe to my channel so you get updates straight away when I post these videos, and like the video, share them. Yeah, do what you like, but thank you so much for supporting me, and I'll see you next week. Bye.